The first part of our winter wildlife webinar series for 2026. My name is Maria Covey. I'm a planner with the Tug Hill Commission. And for anyone that's unfamiliar, the Tug Hill Commission serves a region of about 2,100 square miles of land in parts of Jefferson, Lewis, Oneida, and Oswego counties. The commission's a small, non-regulatory New York State agency that provides technical assistance in areas of land use planning, local governance, community and economic development, and natural resources management for municipalities in the region. Tonight, we'll be hearing from our speakers, Todd Bitner and Daniel Weitoish, about the conservation efforts that they are a part of to restore the ash tree in different New York forests in ways that people like you and me can get involved. So before we get started, I just want to give a brief introduction for our speakers and just go over some logistics for the webinar tonight. So Todd is the Director of Natural Areas for the Cornell Botanic Gardens. He leads in the protection and stewardship of nearly 3,700 acres of natural land. He also lectures in the School of Integrative Plant Science and engages students and the public on natural heritage and conservation issues. In 2020, the Center for Plant Conservation recognized Todd as a conservation champion for his work. Daniel has been with the Cornell Botanic Gardens since 2014. He's held multiple positions and currently serves on the arboriculture as an arboriculture supervisor. He previously held roles at the Morris Arboretum and Scott Arboretum, both in Pennsylvania. He's also an International Society of Arboriculture certified arborist with a tree risk assessment qualification. So during the webinar, we're just going to ask that any questions you have, please put them in the chat and I will summarize them at the end. We'll leave some time so the speakers can get to all of your questions. The webinar will be recorded and we'll be posting it on our YouTube channel in the coming days. So now I'm going to turn the presentation over to our speakers. We're very excited to hear from you.
Great. Thank you for that introduction, Maria. Let me share my screen here and we can get started. The presentation tonight, we're happy to be here to talk about our ash conservation work through the Trees in Peril program. So that'll be part of the presentation, and then also what you can do to manage your ash trees that you have on your own property. And Daniel and I are going to be co-presenting, and he will start. So take it away, Daniel.
All right. Getting into a little bit of an understanding about the insect that we're going to be talking about, the emerald ash borer, this Agrila species, is right around a half inch. We can see that beautiful emerald wing coat in the left picture, and on the right down at the bottom, these are the feeding larvae. This is where most of the damage is going to occur for our ash trees. So you'll hear it a couple times tonight when you hear these S-shaped galleries. This is what we're talking about, the feeding galleries in the living part of the tree. Next slide. Just a quick primer on the life cycle. Generally, pests, they can have one brood or two broods per year. Emerald ash borer almost always has one brood per year. And we can see some of how this life cycle operates up here at the 2 o'clock area. You have eggs that are laid. Through October to March. This is when the eggs have hatched. Larvae are feeding on that living part of the tree, consuming parts of the cambium. They'll feed through much of the spring. April, they will pupate. Sometime in late May, they'll begin hatching and trying to make their escape from the tree. We've got a good picture in a following slide of one of the little buggers trying to make its way out. Next slide. Some of the distribution for this insect. What we're seeing in yellow here, this is confirmed areas that have detected it. In a lot of places, you could have emerald ash borer feeding and growing in for 2-3 years before there's even a detection. So where we're seeing that there's these blank spots, it's not necessarily a lack of emerald ash borer, but rather a deficit of reporting or detection. Next slide. How do we detect? These are small insects. Again, about a half inch. It's very hard to catch an adult. I think I've seen one adult on a leaf since they arrived in Ithaca. Um, so we're looking for other signs that can indicate their presence. And our local woodpeckers are one of the first helpers that we have. You're going to see this characteristic flaking of bark on what would otherwise be that traditional green and white ash diamond-shaped bark pattern. The woodpeckers start flaking the bark off. They are detecting that these larvae are feeding inside of the trees, and they're trying to go after them. Um, Two other things that we'll show in the next two slides are canopy dieback and sprouting that will come from middle of the canopy or elsewhere on the trunk. So we can see that epicormic growth in the left picture here. This is a canopy retrenchment. The tree is suffering. It is not getting the water and nutrients to the top of its canopy because, again, that living part of the tree is being consumed. And over here on the right, these base sprouts, these are both pictures of trees that are incredibly stressed. And while there are other things that could cause a tree to exhibit this kind of stress, this is really characteristic for an ash tree. And keep this in mind too, as we get to the end of the presentation and we're talking about when can we consider treatment, or is a tree too far gone? Both of these are examples of trees that are just too far gone for any kind of treatment intervention. Next. And a close up again on the left here. This was a insect on campus. We caught it trying to get out of its hole. I think this is year two of when we had detected the infestation. And over on the right, this is another close-up of those S-shaped galleries. Underneath the bark, the larvae are feeding and consuming the parts of the tree that will transport both water and nutrients. Next.
Great. Well, thank you, Daniel. So we just kind of wanted to provide a quick primer on some of the basics about emerald ash borer before we jump into the work that we're doing to help conserve ash trees. So about context for that, the work that we're doing is funded through a program called Trees in Peril. And it isn't just about ash conservation, white ash, black ash, green ash conservation, but also trying to conserve hemlock from against hemlock woolly adelgid and American beech for both beech bark disease and now beech leaf disease, forest pests. The work really was inspired by conservation work that started in Vermont, trying to conserve elm from Dutch elm disease. And but in order for this program really to kind of take off, it needed some foundational research. And that work was carried out by some Forest Service scientists, Kathleen Knight and Jennifer Koch, that really asked hard questions and went out and got the science to confirm that we actually have ash that has some level of resistance to emerald ash borer. That science then led to support from a private foundation through the Nature Conservancy with ongoing collaboration with the Forest Service that then kind of rolled up this Trees in Peril program that are working on the conservation for these 5 species. So we have a significant component to that program, but it goes beyond just the work that we're doing. It's, again, some of this foundational science. There's public engagement, there's extension, there's genomic work, there's work on understanding the more about how these trees are resistant and validating that they are resistant, and then onto the breeding program that we're more involved with. So what is a lingering ash? Daniel gave you examples of things that are not, just in the prior slides. So a lingering ash is a tree that appears relatively healthy, like the one in this photograph, in the midst of really significant decline from all the ash tree neighbors that are around it. So essentially the needle in the haystack, one that looks unaffected by emerald ash borer, it wouldn't have canopy dieback, it wouldn't have the significant blonding or the epicormic sprouting that Daniel mentioned. But then you see in this photograph that most of these ash trees around it were healthy and mature, and they've succumbed to emerald ash borer. And so those are the things that we're looking for. Now, obviously, you can't only survey for this in the growing season. And you need to be in an area that has essentially 95% or nearly 95% stand mortality. So that's all the rest of the trees in the vicinity are dead or dying. And they have to be a certain diameter in size because emerald ash borer don't like to lay their eggs on small little twigs. They won't be able to complete their life cycle in those, and so they need some of the more mature trees. So we have a minimum threshold of 8 inches at diameter breast height in order to confirm that it is a lingering ash. And then I put in here a link, and we'll talk about this more. If you do know of a or suspect of a lingering ash tree, how you can document that through one of our conservation partners, the Ecological Research Institute, ERI, and the website monitoringash.org is a place that you can learn more about lingering ash and the criteria, but also for reporting. And then the last point that I want to make here is what lingering ash aren't. And so you notice this is a photograph of the ash in a natural stand. We get a lot of reports from folks that are in suburban areas, urban areas, and they say, hey, here's a tree that's still in good condition. No urban or suburban planted trees can qualify because we're not able to validate all the neighboring dead native genotype trees in that particular area from trees that are in suburban landscapes. So, unfortunately for folks, those are the ones that they see most often. And if they're still healthy, it's more just a lag of them not succumbing yet to emerald ash borer as opposed to them being lingering. So, and some of you might be asking, well, wait a minute. I was told however many years ago that all ash are susceptible to emerald ash borer, and so there would not be such a thing as resistance. And that's actually the common knowledge, the prevailing wisdom for a long time until Jennifer Koch and Kathleen Knight did some of that data collection that identified that this is the case, that we do have lingering ash. But what was happening originally was there was a bias, again, from people looking at trees in urban and suburban landscapes, and they were also coming to emerald ash borer. Well, what we found out was those trees really came from only about 24 genotypes or parent trees. And so the genetic variation in those trees that were in the nursery trade was very, very limited compared to the natural variation that we have in our native forests. So it kind of biased us for a really long time and created this false narrative that all ash trees were doomed to emerald ash borer. And so we know that's not the case now. So how does the resistance work? So there's two uh, theories here. One of them is fairly well documented, which is the one that we're looking at now, that the trees are able to wall off, compartmentalize the cambial layer where the emerald ash borer larvae are tunneling, are burrowing and eating. And so the Forest Service did this study where they intentionally put emerald ash borer eggs into the tree. So you see that there in the second to left photograph. And then they watched the progression of the larvae over time. And the trees that didn't have these lingering um, genes and characteristics succumbed to the emerald ash borer, essentially girdling the trees like we saw in the photos earlier. But the healthy lingering ash trees actually the um, walled off the larvae and killed them prior to them fully completing their development and being able to hatch as adults. So in the middle slide, where the healthy larvae, you can see the tunnel continues, and the blue circle there shows the third or fourth instar larvae that is more mature. And then the lingering ash photos on the far right show that the individual larvae were killed at a much earlier stage and not able to continue and to girdle a tree. And um, and so essentially they're getting walled off and not able um, to continue to eat. So they can't go backwards. They can't go out of the tree or left or right if they're all kind of walled in. And so they essentially starve to death before they're able to girdle the tree. And that's the first of the two mechanisms at work for these lingering ash trees. The second one hasn't been fully proven yet, but is got some pretty strong evidence that it also is um, helps ash trees survive. So all trees, all plants give off these volatile organic compounds, and ash trees give off about 2 dozen. And they are from the normal respiration and photosynthesis that they're creating these chemicals. The emerald ash borer adults um, use those chemicals to find the ash trees to then lay their eggs on. We're not sure which of these yet, and that's part of the proof of concept still on this. We're not expecting that it's all 24 of them. It might just be a handful of them. And that the lingering trees are either producing them at a lower level than the ash borer can detect, or um, maybe at a time outside of the window where the adults are present. So either earlier in the year or later in the year, or maybe a combination of those things. And therefore, they're chemically invisible to the emerald ash borer, and they can't be found to lay their eggs on. We're hoping that both of these mechanisms are true, and that in the future we will be able to use genetic information to identify which trees have each of these characteristics and breed trees that have both of these and create more fit individuals than presently are by themselves. So that's a bit of what is the science behind allowing these trees to have some resistance to emerald ash borer. Now, a bit on the work that we're doing as New York State's Lingering Ash Propagation Center. So, our goal is to um, bank the genetic material for white, green, and black ash for 50 to 60 genotypes, or from 50 to 60 parent trees with duplicates of each, 3 to 5 ramets or duplicates of each, in order to preserve that genetic diversity long term and provide that material for future analysis and studies. And presently, our lingering ash is coming from the Hudson Valley and the Finger Lakes region. And I'll talk a bit more about our larger goals beyond those regions here in a minute. So, a bit on the process. So, first, lingering ash are identified by citizen scientists or other researchers. They're validated by colleagues, and then we're assuming that this tree now is documented and affirmed to be a lingering ash. We'll go out in the winter and we'll collect approximately 10 healthy terminal twigs per individual, preferably from the upper canopy, which is healthier than down in the shaded lower canopy. Collect a whole bunch of information on the site itself, the tree, the conditions, its size, its geographical location, that information stays with that material and all the future propagules going forwards. Um, and and then the last point I want to mention about this is some folks might be like, well, why, if this lingering ash tree is surviving emerald ash borer, why do you need to go to all this work to collect the twigs and propagate them? So the lingering trees are able to persist with emerald ash borer in the environment up to a point. And when you get to this threshold where most of the ash in that particular forest stand start to die, the emerald ash borer population is still really high. It's at this kind of peak level in its population growth. But there's hardly any ash left for it to lay their eggs on. And so the lingering ash disproportionately get targeted in that late phase and essentially get swamped by the millions of emerald ash borer that are still out there. But the thought is after our ash population crashes, then after the emerald ash borer population crashes, doesn't go to zero, but it goes way down, that lingering ash in the future will be able to tolerate that level of emerald ash borer in the environment. But they just can't tolerate it at peak EAB levels, which is why there's this race between getting to the state of being able to collect material and identify lingering ash and collect material and doing it before even these lingering trees succumb in the environment. And so we try to hit that window. So I mentioned earlier that you need to have about 95% stand mortality in order to be in that window. And so the Hudson Valley for the past few years was in that um, at that level. And so we got a bunch of material collected from it. Now it's pretty much past that. And so there's very limited material left there. Our region in the Finger Lakes region is now in that threshold. And so more collections are being made in the Finger Lakes region as we are in that window. The Tug Hill region is not in that 95% stand mortality level yet, but will be shortly. And some areas may already be there, particularly along population centers, parks, campgrounds, along highways, places like that. And so there may be areas that are able to identify lingering ash in the Tug Hill Plateau region now, but if not now, very soon. And and so what we are actually focusing on, we've made huge progress on getting a lot of white lingering ash into our collections, into our conservation bank. We have very limited black and green ash. A large part of that is because it's much less abundant naturally on the environment, in the environment. And so this is where you could help us through citizen science, helping us find more black and green lingering ash. And I'll give you some more resources later in the talk on how you can do that. So now we've collected the scion. Simultaneously, we are growing rootstock to be able to graft that scion onto. It's commercially sourced. We get it as small seedlings. We grow it up in our plant production facility and baby it, put it on drip irrigation, and it's propagated by our amazing staff here at the Botanic Gardens. And then with a partner, Ted Hildebrand through Coldwater Pond, we graft the scion onto that rootstock. And so I'll explain this a bit here. I'll get out my little laser pointer. So here's our tree band, and the roots and the soil are in here. And here is the green ash rootstock. And then we cut it off here. And then we graft on this 8 to 12-inch long stem of our lingering ash scion onto that. And it's wrapped and taped together. And then this whole area here is maintained around 38 degrees because this is happening in January, February, March. We don't want the plants to break bud and to grow and desiccate and all those kinds of things. But ash is notoriously difficult to graft in comparison to things like fruit trees. And what aids the graft is it healing and callusing over, and it does that better when it's actually in warm growing conditions. So inside of this box and sand are two electrical lines that are on a thermostat, and they're kept at 76 degrees for about 8 weeks. And this is called hot callus pipe grafting. And so those are the pipes that are in the sand. And the sand is basically just a buffer so that the temperature doesn't modulate too much. And then we cover it with sphagnum moss. It's kept moist so it doesn't desiccate, covered with an insulation board, and it essentially um, grows inside that very small heated section without either end of the rootstock or the scion growing during that 8 weeks. So here we are, 8, 9 weeks out, and the photo on the left, you can see, I get my pointer again. Here is the grafted scion coming into the rootstock here. And this is where the rootstock is starting and the scion is starting to callus over. So essentially the woody parts are growing back like trees do and heal over wounds. And so it's essentially healing over a wound by healing that and closing that callus. We keep it wrapped in tape because it's fragile and it could still break very easily. And so we then have it in a cold room through late April. And then as the temperatures in the photo period progress, it starts to break bud. So here's a black ash, and here's the bud on the scion that's budding, that's breaking bud, and these up here look nearly to be breaking bud. So that's what we like to see. And here we are a few days after that. And this was our first cohort of grafts in 2023, mostly leafing out there. And from there, then we bring that to our plant production facility. So here we have multiple things going on. In the far background, we have 2-year-old material that was grafted, being grown up on our drip irrigation systems. And that's the material that we planted this past October. And then we have in the middle sections, we have 2 different years of and sizes of rootstock that will be used for future grafting. And then in the near area was the material that was grafted in 2025. We started off in 24 with 150 total grafts, and then in 2025 we upped that to 250, and that's our goal going forwards is 250 individuals from 50 genotypes every year until we meet our conservation goals. We do have some losses over time, and to compensate that, because we want to have a certain number of replicates, we will do something in the summer called a bud graft. So we will go and borrow a bud from one of our grafted trees and put it on rootstock to try to grow a tree from that bud as yet another replicate. Our success for that has been not that great so far, but it's one of the things that we're trying to improve because we, again, want to make sure that we have at least 5 individuals of each genotype represented. So I mentioned some of the genetics before. In this whole process, we're taking leaf tissue samples. We're sending it to colleagues that are involved in the Trees in Peril program, at the University of Tennessee, Knoxville, and they are doing genetic analysis on all the material for all these species so that when we do identify the genes that confer resistance, we can then look on an individual by individual basis to identify which individuals we have which genes. And we also might find that some of the ones that we thought were lingering ash actually may not have either of those resistance genes. And that means that we might divest of those individuals if they don't seem likely to be lingering. So this is where we are presently. White ash is on the left, green ash in the middle, black ash on the right. Our goal is 50 to 60 genotypes, and between the first and second years, we will have met that goal for our white ash conservation bank goals. And then we also have some genotypes in there of what are known as susceptible controls. So those are trees that are known to be susceptible to emerald ash borer that are being propagated for comparison purposes in the future. But you see that we only have 10 and 9 of green and black ash towards our 50 to 60 genotype goal. So that's where we definitely can use some additional help finding more of those individuals in the landscape. So we talked about the propagation side of things. Now we're moving towards installing these individuals into our conservation banks in the field. And so not surprising in New York State, there's deer problems, and these trees are very valuable, so we want to make sure that they're not going to be damaged by deer. So we built some deer exclosures for 2 of our different planting sites here um, in preparation of outplanting, which we did this past October. This is just a snippet here of some of the data that we collect. Each individual is given a unique numerical combination of numbers and letters for tracking purposes. Then we keep track of species, where the material came from, how we propagated it, what the condition was, and all kinds of other data that may prove useful going forward. And this information is connected in the database to these tags that go on each individual, custom tags that have some of the information so that we make sure that we don't mismatch important information that's needed to follow the tree's fate over time with the actual tree in the field. So now we're preparing to actually do our planting. And because some of these species, particularly black ash, have very specialized habitat requirements, we did quite a bit of work to come up with a way to identify what sites had hydric soils that would support black ash, and then how we would randomly assign where planting locations would be for these individuals. And we wanted to do it in this randomized block design so that if there were some environmental variables that contributed to tree mortality, those things would be minimized because we randomly located the trees in the environment, and we might be able to tease that out from no, they failed because they actually are susceptible to emerald ash borer, or that they died from our propagation methods or something else. So we use that to randomly assign our planting locations, and here's the ones that we did in this particular field site for this past October. Then we progressed to marking them out in the field using our rented equipment to dig our holes, bringing our lovingly managed trees for 2 years out to our planting sites, adding compost and vole protection. The little plastic tube there you see is vole protection, and the blue ring is the accession tag, putting out weed fabric around each of them. And voila, we got our first lingering ash planted this year in our conservation bank. So where does that go from here? So now what part of this story? So we're the first conservation bank for New York State. There's interest in creating duplicate banks for New York State. We just saw with the winter storm that we had. Fortunately, we didn't get ice like they did down in the mid-Atlantic, but something like that could really wipe out a lot of work and an important genetic material that we're trying to conserve in our banks if all of our eggs are in that one basket. So creating duplicate conservation banks with propagated material is part of the next phase that we're working on. And then our bank site is meant to be for genetic preservation. That material will, once it's large enough, will become donors for taking scion cuttings and grafting those onto rootstock, and then growing those up and planting those out into field trial plantings. And those trials will be where we actually test for lingering ash. That's done through field bioassays. So essentially grown up to a certain size, protected with pesticides, and then allow the EAB to attack those individuals. And at what point in time the susceptible controls succumb to EAB, the top performing lingering ash kind of make the cut, and those are now the ones that are going to be conserved and used for future breeding and reforestation work. So that's the select the best performing 25%. So from those material, then we could either cross individuals that have the known resistance mechanism, one or the other, or intentionally cross between the two, if we're at that point, between the resistant types, and then collect the seeds, distribute it to different seed regions, create seed orchards, and propagate those individuals, and then grow those up to reforest our forests in New York State. It is a bit of a timeline, but it's very realistic, and this kind of work has been done in conserving other tree species in other areas for a very long time. So it's a proven approach. So how you can help through citizen science. So two different ways. There is an app called TreeSnap. The treesnap.org is there on the page, and you can download that and you can help identify potentially lingering trees, not just for ash, but for a number of different trees that have forest pest issues. So that's one thing that you could do to help us identify green and black lingering ash. And then, as I mentioned earlier, through our colleagues at the Ecological Research Institute, you could go to monitoringash.org, and there's information on lingering ash and how to report it there as well. And we're going to do it. Yay! So with that, I'm going to hand it back over to Daniel and learn more about how you can manage your ash trees.
Outstanding. Thank you, Todd. We've been at this EAB thing for a few years now at Cornell Botanic Gardens, and it's really exciting to have that message of hope in the middle of this presentation. But this is really what we've been talking about a lot. What does it mean to have these ash trees? How do I manage a stand of trees that are likely to die in the next several years? So a little bit about how that happens. Firstly, the center of this, we can see the disruption of phloem. That is the feeding activity of the larva. And broadly, stepping back for a second, it is challenging to identify a singular acute cause for the death of a tree. Usually, we refer to it as this spiral of decline. And we can see from some of the exterior bubbles here, these are the other things that are going to take a healthy tree and slowly begin spiraling down its health. So at the very top, the canopy begins dying back. That takes photosynthetic surface away from the tree. On the left, with the woodpecker damage, there are a few organisms that like additional holes being poked in them. So that can have an impact on its ability to move water and nutrients. On the right, we have root dieback. If the tree doesn't have the roots to get the water into its cambium, that can be a real challenge. And finally, at the bottom, we have the secondary infections. We can see a little bit more of that on the next slide. Starting on the left, canker pathogens. These are pathogens that once there is an entry made into the living part of the tree, they're going to start consuming the tree. At the top, we can see there Cytospora. If for many of you, if you have a spruce tree and it's really suffering, Cytospora canker is in the same family as the Cytospora that would be on ash. So further compounding the living tissue. In the middle, this will be important for our next couple slides, but these wood decay fungi, once they get into the wood of the tree, they begin breaking down different parts of the wood. The bottom, it suggests that, these are mostly white rot fungi. That means that they have the special ability to consume the lignin that is a part of the wood. This lignin is you can think of lignin. I think I have a slide for this after. We'll talk a little bit more about lignin. And on the right, the entomopathogenic fungi. These are kind of exciting. These are fungi that could potentially be used for biological control of the emerald ash borer insect. They colonize an insect, and it frequently leads to mortality of that insect. Next slide. So yeah, a little bit about that wood. We're going to be talking about tree removals in a moment here. So knowing some of the structure of the wood and the impacts of these white rot fungi can inform some of the safety implications. On the left, we have our regular wood. It's an assembly of lignin and cellulose. The lignin, that is kind of like your concrete, and the cellulose is kind of like your rebar. So the lignin is giving this structural, compression resistant structure, and the cellulose is what gives something flexibility. Generally, fungi will specialize in one or the other, or a couple other associated parts of wood structure. But for these white rots, they are removing the rigid part of the wood. Next slide. Um, so what does it mean for you? You have a home or a playground, there's ash trees nearby. How do we go about tackling this issue? The first thing you can do is nothing at all. That is a strategy that surprisingly, not at all, doesn't work out super great. There are instances where you can ignore an ash tree. We have an adage in tree risk assessment that if there's no target, there's no hazard. So an ash tree in the woods, as long as you don't have a tree stand in it, or your trail goes right nearby, that tree you can just let succumb, or see if it is potentially a lingering ash in some future. A more common thing is going to happen regarding removals. We know these trees are going to die, so cutting them down often early is a suggested strategy. I had mentioned that those white rot fungi are consuming lignin. This impacts not only the quality of the wood, but its ability to hold a hinge. If you felled a tree, you know that having that hinge wood gives you the direction and control that you need to put the tree where you want it. Well, we've found that it's harder and harder to trust the wood the further along an infestation has moved. And often there is a period in time where both our arborist teams and arborist contractors locally just said, we're not going to climb any more ash trees. We're done climbing. So it's either a fell or it's a bucket removal. Three, as a hybrid removal, we could, if there's a bucket truck, knock the top out of a tree. That leaves the stem standing, short enough such that it's not going to impact a utility line or a structure. And then that stem can be habitat or food for the woodpeckers at least. And finally, four, we have treatment options that involve insecticides. The next slide. Um, the decision that a lot of people had to make, and something that we grappled with a lot, was do we remove the tree? Do we do nothing, or do we pursue treatment? When it comes to treatment, compared to removal, it's the difference between a one-time cost and a reoccurring cost. So trees inherently have a value. They're giving shade for your home. It's reducing your cooling and energy requirements during the hot season. Sentimental value. So a lot of folks are going to put some money towards that. And what is it going to cost? A tree removal, again, that one-time cost, the tree is cut down and it's done. We could compare that with an herbicide or an insecticide application that could be needed to be repeated every year or every 2 or 3 years. The tree health is very important in that capacity. We need a healthy tree with a healthy vascular system that's going to move the chemical into the tissues. Sawyer safety, again, as trees get more and more canopy retrenchment, more fungal decay in the canopy, you have to be cautious as a saw operator. As that tree starts to go over center during a fell, big pieces can break out of the top. And finally, the consideration of chemical safety is very important. Next slide. Yeah, there it is again. Overall tree health, we need a healthy tree. if we're going to be looking at any kind of pesticide treatment regimens. Um, the cost is going to continue to reoccur. I'm not willing to put like a discrete cost of what a treatment involves. In the past, you may have been able to do this yourself. With some new legislation in New York, some of the chemicals are not going to be available to anybody unless they have an applicator's license. And the final thing to consider here are the impacts of these insecticides on non-target species. Next slide. Some of those non-target implications are frequently a result of these bottom three chemicals you're seeing here. These are in the neonicotinoid class, and they're implicated in the colony collapse issues that are happening with honeybees. You're putting a chemical into a tree. That chemical could be translocated to a flower, for instance, that a bee is attracted to. Not really a concern with ash as they're wind pollinated, but that could still be more chemical going out into the environment, even though it was applied to a singular tree. At the top, we have the emamectin benzoate. That's the option that many people in the industry are leaning towards. A big advantage here is that while it does need to be injected, it has a longer residual. The label suggests you can get 2 or 3 years in between each application. Additionally, it's a less mobile molecule. It's more likely to be captured in the tissues of the tree and not escape through other measures quite as easily. Next. Yeah, that's the wrap with the decision making you have to grapple. No management is an option. The longer you wait to take action, though, the more expensive it gets. As I suggested, there was a time when our local arborist, we decided we cannot safely climb trees. So that means equipment has to come in, and that is going to increase the cost of removal. So getting that inventory early, understanding the scope of what you have to battle, and then removing or treating uh, the following.
Well, thank you, Daniel. So I also want to thank some of our supporters here, the U.S. Forest Service, the Nature Conservancy, and Ecological Research Center. And thank you for joining us on the webinar tonight. And with that, Daniel and I can take questions.
See, so far in the chat, we don't have anything right now. I'll give it a minute if anyone wants to pop anything in if they're curious about. Um, let's see. The app that you had talked about, Tree Snap, that was really interesting. So does that go back? Is that a general report to like what groups does it report back to?
Yeah, so it's been developed by the Trees in Peril collaborative partners, and it's something that is just getting rolled out as kind of a beta. And the reports are going to go to subject matter experts within our groups to do the validation, which is kind of how the monitoringash.org reporting is also working right now. And that information that normally goes along with it is um, information about the tree, but also photographs. So you can imagine a few people might misidentify particular trees. We've had more than one lingering ash that turned out to be a hickory, and so there's some easy ways to vet some of them to begin with. But ultimately, before anybody goes out to collect sign from them, those individual trees get a field verified visit. So it's
Mhm.
not, it's kind of a multi-step process for each of those.
Great. So related to that, actually, we have a couple questions in there. So we had someone ask, is there like a way to tell the difference easily between white, green, and black ash?
That's a good question. Um, there is some hybridization that is happening between green and white ash. So that makes that part a bit more challenging. Um, one of the main ways to differentiate these to begin with is that principally white ash wants to grow in upland conditions, and you'll find it in maple, oak, beech, hemlock forests. And at the other end of the spectrum, black ash, which looks very different than green and white, is mostly in forested swamps with muck and hydric soils, and those two habitats rarely overlap. Green has a much broader, it can grow in some of the black ash habitat and it can grow in some of the white ash habitat. And so that one can be not as easy to pick out just by habitat type. But most of the green ash, like its preferred habitat, is along river and stream terraces and areas that are wet, but maybe not a swamp. Um, and uh, and so in the taxonomy side of this, green and white are very closely related. Black ash is very distantly related. It looks quite different. The bark looks different. The twigs are extraordinarily stout. The buds are very large, and so it really stands out. Some of the characters that I used to use to identify white versus green ash have been found to be unreliable these days. So I used to look at the scar that was underneath the bud and whether or not it was flat or whether or not it was cupped. And I've learned now that that's a very unreliable technique. So Daniel, do you have one that you use regularly?
I have a cheat that I use because I'm frequently handed a leaf, and I may not have other qualities to evaluate it on. So the something that's been working really well for me is leaflet stalk length. We did have a picture in the intro slide that showed the compound leaf of ash. That is a singular leaf that has additional leaflets along its rachis. And the stem that the leaflet is attached to the rachis by is shortest in black ash, intermediate in green ash, and longest in white ash. But again, it's just a little tip that when combined with the totality of everything else, like Todd had said, the location that it comes from, bark features, buds, and leaf scar can often get you very close.
That it's actually one of the reasons that we're genotyping this material is to actually identify what species we're dealing with, so we make sure we have green in the green bank and white in the white bank.
That makes sense. Let's see. So we have a couple other questions that have come in. So we have one that says, is the word scion something that's used specifically in this project, or is that a common term in tree propagation?
Is the word scion?
Yeah, sorry.
Yes. So scion is kind of analogous with a twig, but it is actually specific to material that then you want to propagate from.
Let's see. And then, so similar questions here. So one person said, are there any biocontrols being considered for the emerald ash borer, or do we just kind of have to wait for it to go through all the non-resistant trees and then have the tolerant ones survive?
So Daniel mentioned a newly discovered biocontrol with that fungal pathogen that attacks emerald ash borer. So that appears to be a natural fungus from the United States that has now um, found a new host to utilize. We might be mistaken about that, but that's the prevailing wisdom right now. But the Forest Service and a lot of other conservation partners have been working on introducing parasitoid wasps for a number of years. There's been five that have been approved and released. Three of those five seem to be fairly effective. And I sat in on a talk a year ago from folks at SUNY ESF that looked at um, where these biocontrol wasps, parasitoid wasps, had been released and how far from that release site a few years later were they able to be found. And they essentially found that they had moved over 80 miles from, on average, from the release sites. So if you kind of create a map and you draw 80-mile radius circles, around New York State, nearly all of New York State is covered. Now that's all theoretical based on this, but what it's showing is not only are the parasitoids um, surviving, reproducing, but then they're spreading as well. And so the hope is, and I skipped over this earlier in the talk, was lingering ash that either have the individual resistant genes or the combination of the two, along with the parasitoid biocontrol wasps, would be able to allow ash to persist in the future.
Oh, right. That's very interesting. We also had somebody ask if the once the tree is attacked by the emerald ash borer, is there any way that the tree can recover on its own?
Sure. In a hypothetical situation, if you have one ash tree and a singular adult lays eggs, the larvae go through their life cycle and hatch and fly away as adults, and then there are no more larvae next year. The tree is trees are constantly fighting this kind of assault. I talked about some of the fungi and the cankers and all of these things that are constantly attacking trees. They can't get up and move, so they're just at the mercy of what's around them. So they're always in that struggle of um, something consuming them, trying to outgrow the thing consuming them. So yes, absolutely. If the pest pressure went away, the challenge, however, is you can see these graphs where emerald ash borer is detected, and we start with a low population, and the population just climbs, and it gets incredibly high. And that is when you turn around, and in 2 years, you're like, oh my goodness, all the ash trees have died. So it's the constant high pest pressure that's a real challenge.
So I'll add to that. There are some recent discoveries that were reported in some of the areas where emerald ash borer first occurred in Michigan and Western Ohio, and there were mature trees there that were in very, very poor condition, and some of them seem to be regaining some of their health. And that doesn't mean that they're going to persist long term. It's not clear yet exactly why. Do they have some, maybe they were a lingering tree, but then they got overwhelmed by the emerald ash borer population when it was at its peak, but they didn't fully die and now they're coming back. So we're not sure exactly what to make of those yet. Um, and so that's a whole bunch of what ifs around that question of whether or not trees can persist on their own. But we're still, each of these forest pests is novel. And so it's, even though we learned so much from the past ones, you still have to start from square one on every single one because there's some things about them that are unique.
Understandable. So this one's applicable, especially right now, but we have a question about, do the extremely cold temperatures in the winter, can they kill off the larvae from the emerald ash borer?
Oh, we would love it if that were the case. So for some forest pest species, the answer is yes. So for like hemlock woolly adelgid that we care about, um, we see winter mortality with -5, -10, -12. Um, the research out there for emerald ash borer is something like -40. And that part of that is because the insulating properties of the wood. So they're inside the bark, inside the cambium, and that provides a thermal buffer for that. Because it's not like it's negative, even if it's -15, it's not -15 for 24 hours, day after day after day, right? It's -15 for one cold night, and then it gets warmer again. So that's part of why it requires so cold temperatures is that combination of duration and the insulating properties of the wood. So I wish I had better news on that one, but that one's not one for us to hang our hats on.
There was a paper
Understandable.
out of the Midwest that said -35 was pretty impactful. Um, but I don't know a lot of folks that are, fingers crossed for -35.
Right, exactly.
And I have heard that, I talked about some of the life cycle. If you have an extremely cold, enduringly cold winter, that could slow down the larvae's development, and it could take their life cycle 2 years instead of 1 year.
Okay. So slow it down some. Seems to be quite a hardy bug.
Yeah.
All right, let's see. I'm just gonna do, we are at 8 o'clock. It's doing a final scan of questions. So treesnap.org, off the top of your head, do you know if that's the correct website
Yep.
for the app? Okay, perfect. And then you said the white ash are in the drier areas generally.
Yes.
Okay. So I think that wraps up all the questions that we have. Do you either of you have any final words? I really appreciate you speaking tonight.
No, we're happy about the interest. Thank you for the invitation. It's been a pleasure. Um, I hope folks can help us conserve ash, go out and help us find some, and keep up hope, as Daniel said.
Great. Thank you so much. For everybody on the webinar, then I'm just going to do a quick reminder that next month we'll have part two of the Winter Wildlife webinar series on February 26th, also at 7 p.m. That one's going to be focusing on wolverines. We'll hear from staff at Zoo New York here in Watertown, New York. Anyone with any questions is welcome to email me at maria@tughill.org. And with that, hope everyone stays healthy, stays warm, and thank you very much for attending.